Western blotting is a powerful and widely used technique in molecular biology for detecting specific proteins in a sample. It combines gel electrophoresis, membrane transfer, and antibody-based detection to identify proteins based on their size and reactivity. Before we begin the transfer step, let's quickly recap the earlier stages of Western blotting. Proteins are first extracted from cells or tissues using lysis buffers. The extracted proteins are then denatured with SDS, loaded into a polyacrylamide gel, and separated based on their molecular weight during electrophoresis. Now, let's dive into the detailed part, starting from the transfer of proteins from gel to membrane. Once electrophoresis is complete, the next critical step is transferring the separated proteins from the polyacrylamide gel onto a membrane, such as nitrocellulose or PVDF. This is where Western blotting truly shines. We create a transfer sandwich by layering filter paper, the gel, the membrane, and another filter paper. This sandwich is placed between two electrodes in a transfer apparatus, with the gel facing the negative electrode, cathode, and the membrane facing the positive electrode, anode. Since proteins are negatively charged due to SDS, they migrate toward the anode and get trapped on the membrane. The transfer typically runs for 1 to 2 hours or overnight at lower voltage, often with cooling to prevent heat damage. Before applying antibodies, the membrane must be blocked to prevent non-specific binding. This is done using a solution like 5% BSA or non-fat dry milk. The blocking agent covers any unoccupied areas on the membrane, ensuring that antibodies only bind to their specific target. Blocking typically takes 1 hour at room temperature or can be done overnight at 4 degrees Celsius. After blocking, the membrane is incubated with the primary antibody. This antibody specifically recognizes the protein of interest. It is usually diluted in the same blocking buffer and incubated for 1 to 2 hours at room temperature or overnight at 4 degrees Celsius for better binding. After incubation, the membrane is washed several times to remove unbound antibodies. Next, a secondary antibody is added. This antibody binds to the primary antibody and is conjugated with an enzyme such as horseradish peroxidase or alkaline phosphatase. After incubation with the secondary antibody, the membrane is washed again thoroughly to remove excess secondary antibody. Next, we add a substrate that reacts with the enzyme and the visible bands are shown, which tells us our target protein is present and how much. Don't forget to like, subscribe, and comment if you found this video helpful. Stay tuned for more tutorials.